Rahim. Assalamu alaikum. This is Dr. Huri Amir. Now, today's lecture is regarding the muscle sensory receptors. Now, what are muscle spindles? Muscle spindles are distributed throughout the belly of the muscle and they send information to the nervous system about the length of the muscle, the rate of change of length of the muscle, or the stretch receptor. The other sensory receptor present in the muscles, that is the Golgi tendon organs. They are located in the muscle tendons and transmit information about tendon tension or rate of change of tension. They are also given the name of tension receptors. Now look at this diagram. You can see the receptor function of the muscle spindle. You can see that there is presence of the muscle spindle, there is presence of Golgi tendon organs, and the proprioceptors, spindle fibers, they are going to sense stretch, and that is going to be taken up to the sensory nervous system by active sensory neurons, and then by gamma motor neuron and the other motor neurons, they are being given the information for motor activity. There is the microscopic structure of muscle spindle receptor in detail. Now, what is this structure? This is actually having this shape of a spindle. You can see the shape of a spindle, which is about 3 to 10 millimeters long. It is built around 3 to 12 tiny intrafusal muscle fibers, which are pointed at their ends and attached to the glycocalyx of surrounding large extrafusal skeletal muscle fibers. The sensory innervation of muscle spindle is that the receptor portion of the muscle spindle is its central portion. The intrafusal muscle fibers, they do not have myosin and actin, which are the contractile elements in the central portion. So, the microscopic structure of a muscle spindle, you can see that is actually a spindle shaped structure which is having the length of about 3 to 10 millimeters, and it is lying in the central portion of a muscle. And over here the contractile proteins, which are actin and myosin, they are absent. Now, how the excitation of muscle spindle occurs? It actually occurs by two ways. That is, the lengthening of the whole muscle stretches the mid portion of spindle. That is, the receptor excites. Then, the contraction of the end portions of the spindle's intrafusal fibers stretches the mid portion as well. Although the length of muscle does not change, but the receptor excites. Now, the central receptor area of the muscle spindle consists of two types of endings, which are primary endings and secondary endings. Now, primary endings, what are they? A large sensory nerve fiber encircling the central portion of each intrafusal fiber in the center of the receptor forming. They are the primary endings or annulospiral endings. The nerve fiber is a type 1A fiber, 17 micrometers in diameter, and it transmits the sensory signals to spinal cord at a velocity of 70 to 120 meters per second. Now, this is actually the mid portion of the muscle fiber. You can see that there is presence of muscle spindle and how the innervation is taking place, What are the primary fibers over there? For efferent nerve supply, we have secondary fibers. Then for efferent fibers, we have the gamma fibers and alpha fibers. The afferent nerve fibers, they are meant for the sensory supply, whereas the efferent nerve fibers are for the motor supply. So you all are supposed to draw the structure and draw the structure of muscle spindle, showing both the efferent as well as afferent supply. The secondary endings, usually one or two smaller sensory nerve fibers, type 2 fibers with 8 micron meter diameter, innervate the receptor region on one or both sides of the primary endings. This sensory ending encircles the intrafusal fibers as type 1A fiber, but it spreads like branches on a bush. Secondary endings. Then we have the intrafusal fibers, which are divided into two types of fibers, which are nuclear bag muscle fibers and nuclear chain fibers. Now, what are the nuclear bag fibers? 1 to 3 in each spindle. Many muscle fiber nuclei are congregated in expanded bags in the central portion of the receptor area, whereas the nuclear chain fibers are 3 to 9. They are about half the size of nuclear bag fibers, and they have nuclei which are aligned in a chain throughout the receptor area. You can see the nuclear bag fibers. There is presence of nuclear chain fibers. The primary sensory nerve ending, 17 micron meter sensory fiber, is excited by both the nuclear bag intrafusal fibers and the nuclear chain fibers. The secondary sensory nerve ending, 8 micron meter sensory fiber, is usually excited only by nuclear chain fibers. The gamma motor nerves to the muscle spindle can be divided into two types, which are gamma dynamic, gamma D, gamma static, gamma S. Now, the gamma D excites mainly the nuclear bag intrafusal fibers, whereas the gamma S excites mainly the nuclear chain intrafusal fibers. Now, when we are learning a lot about muscle spindle, the efferent supply, the afferent supply, we should be knowing about what is this term static response. When the muscle spindle stretch slowly, both the primary and secondary endings continue to transmit their signals for at least several minutes if the muscle spindle itself remains stretched. Stimulation of the gamma S fibers enhances the static response while having little influence on the dynamic response. Number of impulses transmitted is directly proportional to the degree of stretch. Then there is a dynamic response. Now, what is dynamic response? Length leads to primary ending stimulation powerfully, not the secondary ending. This excessive stimulus is known as dynamic response. Primary ending responds extremely actively to a rapid change of length in spindle length. When the spindle receptor shortens, exactly opposite sensory signals occur. The primary ending sends extremely strong, either positive or negative, signals to the spinal cord to apprise it of any change in length of the spindle receptor. When the gamma D fibers excite the nuclear bag fibers, the dynamic response of the muscle spindle becomes tremendously enhanced. Muscle stretch reflex. The simplest manifestation of muscle spindle function is the muscle stretch reflex. Now, muscle stretch reflex is the beautiful example of a monosynaptic reflex. Muscle stretch reflex is frequently asked in exams. The sudden stretch of muscle leads to excitation of spindles, reflex contraction of the large skeletal muscle fibers of the stretch muscle, and also closely associated synergistic muscles. You can see a monosynaptic reflex, how it is occurring. Now, we are going to talk about the classification of reflexes. Now, reflexes, they are of different types, and broadly they are being classified into monosynaptic reflex, polysynaptic reflex, and visceral reflex. Now, the examples of monosynaptic reflex can be a muscle stretch reflex or tendon jerk, like bicep jerk, triceps jerk, knee jerk, ankle jerk. Then we have polysynaptic reflex, which is including multiple synapses, and this is like, for example, withdrawal reflex, abdominal reflex, plantar reflex. Then we have the visceral reflex, like for example, micturition reflex, defecation reflex. Then we have the anatomical classification of reflexes. We have segmental reflexes. In these, end of efferent neuron and beginning of efferent neuron, they are lying there in the same segment of spinal cord. That is why the name given to them of segmental reflexes. For example, the reflex arc that is passing through only one anatomical segment. And what is that? For example, knee jerk reflex. Then we have the deep reflexes. The stimulation of the receptors deep in the muscles, they are being involved, and they are basically stretch reflexes. They are also called as tendon reflexes. For example, there is ankle jerk, knee jerk, etc. Then there is visceral reflexes. Are the reflexes where at least one part of reflex arc is autonomic nerve. So remember, what are the visceral reflexes? That in visceral reflexes, the one part of reflex arc is always an autonomic nerve. There is stimulation of receptors in the viscera. For example, carotid sinus reflex, then pupillary reflex, etc. Then we have pathological reflexes. These are actually the type of reflexes which are not present normally, and their presence actually indicates that some pathology or some disease is there in the body. For example, Babinski's sign. That is one of the beautiful example of pathological reflexes. Now, what are the components of reflexes? There is always a sensory receptor, then sensory neuron, then integration center, then motor neuron, then effector. So these are the 5 components of reflexes. See, over here, there is always a sensory receptor, number 1. Number 2, you can see sensory neuron. Then number 3, there is an integrating center, which can be spinal cord or brain. Then there is motor neuron, and last but not the least is the effector. which can be a muscle or a gland, and it contracts and relieves the stretching.